Now, the term DNA replication refers to the process of copying a DNA molecule. So this is a process which was best understood by watching the animation, and I do have the link to the animation on the web study site. You can always find some great animations on internet. Your book has a great one too, and I'm going to show it in class as well to go over it. Too bad I cannot show an animation using this software and also having to record. So I'll sort of go briefly up here because you really will get a better feel of this when you watch an animation. The first thing is Watson and Crick use the term semi-conservative to explain the DNA of replication process. Now, the term semi-replication is used because one of the strand is the old strand, which is conserved or present in the new daughter cell, and the second or the complementary strand is the new strand. So for example, up here you can see how the double helix is shown, and the blue strands is the old strand, which is now separated more like a zipper. So if you can imagine a zipper being opened up and how the template is acting on both sides as the old strand, and the new strand is then being added as a new nucleotide sequence, and this is the reason why it's called as semi-conservative. Now, there are three stages to the replication process. The first stage is the most important, which is the unwinding. The unwinding stage means that the old strand makes to have a nick somewhere in the strand, and then that nick or the breakage of the hydrogen bond between the paired molecule is the most important part, and that that takes place by an enzyme called as helicase. Helicase is an enzyme that will come and recognize to break the first hydrogen bond between the two paired bases. And there's actually another molecule which we are not going to talk about it, but there are protein binders which actually keep the two strands pulled apart. Otherwise it can come back and rejoin the bonds. So the book doesn't explain it, but when you watch the animation it will show it to you. Now, once we have done that, the second part is the complementary base pairing. Now, the complementary base pairing needs these another kind of enzyme to help construct this, and this other enzyme is called as the DNA polymerase. A DNA polymerase is actually going to be present in step 2 and later also into the joining process, but it works in a classic example which is done within a lab setting, or as we call it, in vitro experiments. The backbones of the parental DNA molecules are shown in the bluish region, and the new ones are in green. Just keeping that in mind. Now, we'll talk a little bit later about the leading versus the lagging strand. I'm going to spend a little time on the next slide talking about it. But before we go on, what does the DNA polymerase do? DNA polymerase is going to find the nucleotides that must be present as a pool. So you should have a pool of adenine, guanine, cytosine, thymine nucleotide, and it will come and pair up accordingly. So for example, it's kind of hard to see the first one, so let's go ahead and look at it over here. You can see that this is C. So if this is the C which is present in the old strand, the DNA polymerase is going to find the nucleotide G and bring it so it can bond hydrogen bonds with the cytosine, and then go ahead and read the second template, which is C again, find another G, bring it up here, keep on working, goes and reads the third template, which is T, and picks up another nucleotide A. So it's aligning these nucleotides, and as they are aligning, it also helps form these connections between the nucleotides, so the nucleotide is also growing as it's doing that. And eventually you will get a long new strand which is complementary to the base strand. So the ligase is a third enzyme which helps in joining these nucleotides. So we have examples of three enzymes here, three major enzymes that help in the process of DNA replication. So helicase is an enzyme that takes place during the unwinding process. Then we have the DNA polymerase, which will actually be present in both cases, but it actually helps in reading the template and bringing the nucleotide. And the third enzyme, which is the ligase, which joins the nucleotides. So now that we've understood the three different enzymes and their role, we will now go into a little bit more depth of the leading versus the lagging strand. Now, the leading and the lagging strand is important to understand because the DNA template is going to be read only in one direction. Okay. Now, during the replication process, the DNA polymerase has to synthesize the daughter strands in the 5' to the 3' direction only. So the DNA polymerase is going to go in this direction only, 5' to 3' only. Now when the two strands are open, notice you have a 3' end on the blue here and a 5 on the blue here. So which means that the DNA polymerase can make one chain only in the 5' to 3' end, and it can go continuous because that zipper keeps on opening. The DNA polymerase is reading the template and bringing the nucleotides, and it can go in a continuous manner, which is the leading strand. However, recall that the two strands were anti-parallel. So we have 5 and 3. So if this is going to be a continuous chain, because it's reading from 5 to 3 here, what about this one? This chain is being opened, but you have the 5' end here and 3'. So you really can't make initially any segments unless the chain opens up and then it starts forming a strand in this direction. So this other strand is actually going to be made in pieces, and these pieces are called as the Okazaki fragments, because the Okazaki fragments are actually called Okazaki because Okazaki was the Japanese who discovered them. What he's saying is because of the situation for the replication of the other parental strand, which is running into 3 to 5' direction, which is away from the fork. This is the fork, or the replication split as it's called. The daughter strand must begin from the fork end, which is from the inside out. Therefore, the replication begins over and over again as the DNA polymerase slides on to the main strand. So the leading strand is opening in one continuous direction. The lagging strand is going to be discontinuous, and it will result in these fragments. And later on, the enzyme, which is the ligase, is also going to come and link all these fragments together to give you a long nucleotides. I hope this sort of makes sense. We will watch this as the animation again, and hopefully you will have a better understanding in depth of the DNA replication. Some of the pieces of replication, again, I'm going to summarize up here, is the structure of the deoxyribose nucleus has nitrogen bases, that is the adenine, guanine, cytosine, thymine, and the phosphate sugars are all present on the stems. Now, the template strand and the replicated strand will be formed after we understand the term semi-conservative, because one strand is going to be the template strand, which is the original strand, and one would be the new replicated strand. The replicated strand can only grow from 5 end towards a 3 end. Therefore, before the replication begins, an enzyme, which is the helicase, will separate the DNA strands, there are proteins called as the binder proteins that hold these two strands separately, and the replication will continue in a continuous process in the leading strand, but a discontinuous process for the lagging strand. In both cases, There is an enzyme which we will see in the animation called as the RNA primer, which will actually add a primer before the DNA polymerase adds the nucleotide to the 3' end, and eventually the two strands will grow. The lagging strand is discontinuous, so an enzyme, which is the DNA ligase, will join all these fragments to form a complete strand. So there are quite a few members of this process. When you come in class, I'm going to have you do an act in which we will have somebody will take roles for all these different enzymes. We will take roles of all the different products that are needed, or the different starting material that are needed for this replication process. So I'm going to go ahead and conclude over here, and again we will reinforce this during class.